Hello friends, today we are going to discuss about nerve impulse conduction along a myelinated and a non-myelinated neuron. Before we go further with the nerve impulse conduction along the length, we must understand the difference between the structure of a myelinated and a non-myelinated neuron. We say each neuron has dendrite, cyton and axon, and the nerve impulse conduction is unidirectional from a dendrite to axon in a way that it goes from cyton to axon. Correct? And this nerve impulse conduction travels along the axon of nerve fibers as an electrical impulse because of the change in the ionic potential along the axon membrane. Now, how does this electron or electronic potential is created along the axon membrane is because of the opening and closing of the ion channels, right, which we have discussed in the nerve impulse conduction. Now, how is it different in a myelinated or a non-myelinated neuron is that myelinated neuron has a wrapping along its axon length of a material, protein material called myelin that is synthesized by Schwann cells. These Schwann cells create or synthesize a myelin coating in the form of a repeated wreath-like concentric layers over the axon, and this myelin sheath now acts as an insulator. That means the area where the myelin sheath is present, there will be no entry or exit of ions from that particular area of the axon membrane across the axon membrane. So, if it is continuously present over the entire length of the axon, there will be no exchange of ions, right? From this point onwards, no exchange of ions will happen. So, if no exchange of ions will happen, there will be no potential difference across the axon membrane. So, there will be no electrical impulse, there will be no conduction. To avoid this and make sure that the conduction is happening, this myelin deposition is not continuous. It's discontinuous. There are certain areas in the middle where the myelin sheath is not present. Those areas where the myelin sheath is not present, my dear, are known as the nodes of Ranvier, where the ion exchange can happen across the nerve membrane. Now, in this case, what happens is, we understand that nerve impulse conduction occurs when there is a threshold stimulus, and upon the threshold stimulus, the sodium ion channels open. Ideally, inside a nerve membrane, it is negatively charged with a potential of resting nerve potential of minus 70 millivolts, and there is a positive charge outside because of excess of sodium. Whenever there is a threshold stimulus, the sodium channels open, the sodium enters inside. That means now the positive charge of the sodium is going to enter inside and make the outside or the extracellular fluid relatively negative, and inside is relatively positive. If I compare the ionic potential of this area to the ionic potential of this area, I would consider that now there are 2 points, point A and point B, with a different potential. If this is point A and this is point B, point A has a positive potential, point B has a negative potential, and there is a difference in the potential. Due to this difference in potential, we say the potential difference creates a force. Like we say, there is an electron motive force because of the difference in the charges when we study current. Likewise, because of the difference in the ionic potential, there is a difference in the charges in these 2 points, which is going to lead to the flow of the electrons from one point to the other point. Now, if the electrons flow only on one side of the membrane, that is inside, say from here to here, what is going to happen? The other side is going to become positive, and this side is again going to become negative. If I compare it with the outside, now inside is also negative, outside is also negative. Above at B point, inside is also positive, outside is also positive. So there is going to be a balance of the potential on either side, and if there becomes an equilibrium, the resting membrane is going to become depolarized, which is not the property. The property of a nerve membrane is that it is polarized. There is a difference in the potential of the extracellular fluid and the intracellular fluid. There is a gradient of the channels, or there is a gradient of the ions, sodium ions and potassium ions inside and outside, which is responsible for flowing of the ions in and out to create an ionic potential. If only the inside ions move and there is no movement outside, I will not be able to again get the resting membrane potential, unless my sodium potassium active pump are working for sure. But I have diffusion channels also which help us to maintain this resting membrane potential. Those diffusion channels will not move the ions because there is no difference. Therefore, it is very essential for the nerve impulse conduction that if there is flow of charges inside, there has to be a flow of charges outside to complete the circle or the circuit and maintain the difference in potential which has been generated because of an outside force, the threshold stimulus. Therefore, whenever we have a nerve impulse conduction, there is flow of charges inside in the intracellular fluid, and there is a flow of charges outside in the opposite direction. If the charges flow inside from point A to B, outside they flow from point B to A, so that there is a polarity difference which is created. If this is negative and positive here, I will move it in such a way that the positive of inside, this is positive because of the threshold stimulus. Now this positive turns negative and outside negative turns positive. Then only I complete the circuit. Therefore, the movement of ions on the outside of the exolemma is also important. And if the outside is coated by a myelin sheath, there will be no conduction, there will be no movement of ion. It is going to act as an insulator from outside to inside or from inside to outside. बाहर की तरफ कोई चार्जेस फ्लो नहीं करेंगे, बाहर की तरफ चार्जेस फ्लो नहीं करेंगे तो मेरा सर्किट कंप्लीट नहीं होगा. And if my circuit will not complete, I will not have an effective nerve impulse conduction along the length of axon fiber. So in the area where I have a myelin sheath, there is no exchange of ions. But in the area where there is no myelin sheath, I have exchange of ions. So what happens when the nerve impulse reaches this point from where the myelin sheath starts? It jumps because this is going to act as an insulator. In the entire length, I would have no exchange, but I will experience exchange at this point where there is no myelin sheath. So immediately the flow of charges happens from point A to point B, which exists here now, not just adjacent but at a distance. And then again it is going to jump to the next point, which is at a distance. In a non-myelinated neuron, it moves from one point to the other which is adjacent to it. That means from point A to point B to point C to point D. They are just very closely placed and adjacent to each other, like taking baby steps and moving along the length of the road. But in a myelinated neuron, the 2 points between which there is going to be the flow of charges are set apart. And it is like a person is taking a long jump or a leap or is saltating like a forward roll or a cartwheel from one point to the other point, so that it travels a long distance in one go. And when it travels a long distance in one go, as compared to the baby steps, it's going to reach faster to the destination as compared to the baby steps. That's why we say that conduction along a myelinated neuron is relatively faster because of saltation. Saltation means jumps, long jumps at nodes of Ranvier, because nodes of Ranvier are the areas where they complete the circuit and there is potential difference which is helping us to flow the charges and conduct the impulse forward. And such type of conduction is thus known as saltatory conduction. of nerve impulse. Whereas, if I compare it with the nerve impulse conduction along a non-myelinated neuron, it happens relatively slowly from one adjacent point to the other along the entire length of the axon of a nerve fiber. Hence, there is a slight delay in reaching the messages from the dendrite to the last nerve ending of a particular nerve fiber in comparison to a myelinated neuron. All the myelinated neuron axons appear white and form the white matter of the central nervous system. If you would remember the structure of a brain, it has grey matter on the outside and white matter in the inside of the brain. The grey matter contains the cell bodies forming the cerebral cortex. The white matter are all the myelinated axons. Clear? So I hope the topic of difference in conduction along the nerve fiber axon of a myelinated and non-myelinated neuron is clear to you now. We will see you with more videos very soon. Thank you very much.